nutrients are required to maintain my body is maintenance requirement. And if I do any sort of activity, if I just stand up, I walk, I talk, so for all that, the requirement comes under the production side. So as if we talk about the dairy animal, the production requirement is of milk production. If we talk about the hens, it will be the egg production. If we talk about the sheep, it is for wool production. So everything except the maintenance comes under the side of production. So there are two different types of requirements, and each and every body of any of the animal is having those requirements. So these requirements are fulfilled through the nutrients. Nutrients like protein, carbohydrate, fat, minerals, vitamins, these are the nutrients, different category of nutrients. So these nutrients are provided from the feed, which is the source of that nutrient. So feeding that source in a proper manner to fulfill the requirement is known as scientific feeding. So this is the definition of the scientific feeding. Now, that what is the importance? So importance may be through proper scientific feeding, we can maintain the health status of the animal. We can uh, recoup the animal from any diseased condition. There the economic production will be there. There the less wastage of feed. Okay, we are having the farmers having more profit. So all these are the benefits of the scientific feeding. Now there are many different types of experiment which we usually conduct in the nutrition, and to evaluate the importance of any particular nutrient, or if we have to know evaluate any particular feed source, then we have to conduct some or the other type of experiment. So all these experiments are conducted for different purposes. So there are different types of experiments. Like first is comparative feeding trial. When there is a comparison in between the two different feed stuff, at that time we perform the comparative feeding trial. So when we have to compare two different feed sources, then we can go for the comparative feeding trial. In this, for one group of animal, we fed uh, one type of feed, and in the second group of animal, we feed a second type of feed, and then we can evaluate the health status of the two groups of animals. That which one group is having better health status, better production, so that feed is having better nutritive value. So this is the comparative feeding trial. We are comparing two different sources or two different feed stuffs, but we are unable to prove the superiority of one feed over the other. Yes, but we can't prove the superiority in terms of the direct feed, because it may possible that that particular species of animal is not the feed which we have fed to that animal, which is good for that particular animal. So if we suppose if we fed the mustard oil cake in case of a poultry, the production performance of the poultry will be reduced because that particular feed is not suitable for poultry. So we can't just judge the superiority of that feed because we are not evaluating the feed. We are just uh, proving that on the basis of the feeding trial. So this is not suitable. Feeding trials with laboratory animals. So results of experiments over lab animal are not implemented over farm animal. It can be used as a pilot study. So as the digestion metabolism of the laboratory animals are far different than that of the ruminants. So uh, we can't judge the performance of laboratory animal and implement it in the ruminants because their performance or their everything physiology is totally different. But that lab animal trial can be used as a pilot study.
सिरदर्दी रहती।
So the next trial is laboratory animal, and as I say that we can't implement the results of the laboratory animal trial over the large animal because they both are different, but we can use it as a pilot study. Pilot study is basically a short-term study so that we can evaluate that particular feed on a smaller basis. So we can perform the experiment on the lab animal and we can evaluate the efficacy at a very small number over the lab animal, and then we can go for the experiment, a large experiment over the ruminants. The cost of experiment is less, yes, on the laboratory animal. The cost of experiment is less because the laboratory animals are cheap and their feeding is also very economical. So the cost of overall experiment is less, individual or environmental variability is very less. So the individual variation in between the lab animal is very less and they can be kept on under a controlled environment. Okay, we can keep all the lab animals under the same one room, so the environmental variability is also less. Slaughter or chemical composition can be performed, so it is easy to perform slaughter experiments on the lab animal, which is not possible in the ruminants. So we can go for the total retention of the nutrients also by the slaughter technique. So these are the benefits of the laboratory animal experiments. The next type of experiment is purified diet method. This is also one type of experiment, that is purified diet, because if we want to evaluate any particular, suppose if we want to evaluate mustard cake, so mustard cake is having the protein also, carbohydrate also, some amount of fat also, minerals also, but all these nutrients are also provided by the different ingredients. Like if we talk about a maize grain, so maize grain is also having some amount of protein, some amount of carbohydrate, some amount of fat, and minerals and vitamins. So all the ingredients which we are including in the feed, all are having some or the other amount of all the nutrients. So we can't judge any one nutrient. So if we want to see any one nutrient or the availability of any one nutrient, we have to go with the purified diet method. And in this purified diet method, we take a purified nutrients. It, it is consists of purified sources of various nutrients such as protein. For protein, we take casein, carbohydrate, we take starch, fat, we take lard or oil, minerals and vitamin, we take pure salt or compounds. So everything is solo is responsible for, is the sole source of the solo nutrient. Casein is only responsible for protein. Starch is only having is a carbohydrate. Oil is only having fat, none other nutrient. So in between this, all these nutrients we can fulfill the requirement of all other nutrients by these purified source, except our one variable, except the one variable, and that is our test product. So this is the purified diet method. Okay. Now the next experiment is germ-free technique. Germ-free technique is usually uh, they are used in the pathological or microbiological research. Although we are also using this germ-free technique, and in this, the intestinal organisms in the host sometimes create complication and interference of the interpretation of the data. So what we take specific pathogen-free animals. Usually, these specific pathogen-free animals are developed in case of a laboratory nowadays. So laboratory animals are SPF, specific pathogen-free animals are coming in case of a laboratory animals. But usually in the large animals or in the small ruminants also, we can't create such type of line, that is SPF line. So laboratory animals are coming and we have to conduct an experiment on the laboratory animal. If we want to go with such type of experiments, we have to conduct the experiment on laboratory animals. So animals were totally free of contamination, that is bacteria, yeast and molds, etc. So all the GI tract microorganisms are, they are free from all the GI tract microorganisms. So none of the bacteria or the mold can interfere with the absorption or the wastage of the nutrients. Group feeding versus individual feeding. Yes, group feeding versus individual feeding experiments are also conducted, in which we can go for the individual feeding of the animal, or we can feed the animals in a groups. We can divide 6 animals in one group and we take the average of all, or we can go with the individual feeding methods. So individual feeding is better than that of the group feeding, because we can get the data of individual animal. There's a, we can avoid the variability, individual variability, if we go into for the individual feeding. In a group feeding, usually the individual variations are there, but we have to suppose that there is no individual variations. If we go for the group feeding, we have to suppose that all the animals are of the same body weight, same age, but this is not possible in case of a large animal. As the animal become larger, the variation, individual variation becomes more. So in a scientific experiments, basically we have to randomize all the animal. Randomization means हम उसका एक ग्रुप ऑफ एनिमल का हम एक एवरेज बनाते हैं. उस एवरेज में हम उसकी मिल्क यील्ड, उसका बॉडी वेट, उसकी पैरिटी, इन सबको कंसीडर में करते हुए हम उसका एक एवरेज बनाते हैं. सो जिस भी पर्टिकुलर ग्रुप्स ऑफ एनिमल का एवरेज नियर अबाउट सेम आ गया, उसको हम कंसीडर कर लेते हैं. लाइक 6 एनिमल इन फर्स्ट ग्रुप, 6 एनिमल इन सेकंड ग्रुप, 6 एनिमल थर्ड ग्रुप. तो जब हम इनका रैंडमाइजेशन करते हैं एनिमल का, यहाँ वहाँ उनको शिफ्ट करते हैं और हम एक एवरेज बनाते हैं. तो जब भी जिस भी कॉम्बिनेशन ऑफ एनिमल्स में इन तीनों का एवरेज एक सा आ जाता है, वी कैन फाइनलाइज दैट 3 ग्रुप्स. तो जहां भी उनका बॉडी वेट का एवरेज, मिल्क प्रोडक्शन का एवरेज और पैरिटी का एवरेज तीनों सेम आ गए, दैट मींस द एनिमल्स वर रैंडमाइज्ड एंड और नाउ द इंडिविजुअल वेरिएशंस आर मिनिमम. Control versus ad-lib feeding. So ad-lib feeding is most common. Ad-lib feeding means जितना खा सकता है वो, ad libitum. It means as much as the animal can eat, it is ad-lib feeding. So ad-lib feeding usually it is very common, but it doesn't give result for certain purpose like digestibility, because for that digestibility we need some data of intake outgo. So if we go for the ad-lib feeding, we never calculate the total feed intake of the animal. So for the calculation of certain parameters like digestibility, we have to control the feeding. हमें उसको एक control feeding में मतलब fixed amount of feed we should offer to the animal, so that we can calculate its feed intake, dry matter intake. In trials, controlled feeding is followed. So in any of the trials, we can go for the controlled feeding of the animal. Equalized paired feeding. In this equalized paired feeding, what we do, we first prepare 2 groups, and both the groups were kept on the same feeding. That is a preliminary phase. In a preliminary phase, we keep the animals on the same feeding, and after that, we after preliminary period, we fed them the test feed. So for 7 days, we keep on the same feed, so the animal performance, जो body weight है, वो अपनी कंट्रोल फीडिंग में, प्रिलिमिनरी फेज की फीडिंग में उसका सेम आ जाएगा. उसके बाद जो हम उसको टेस्ट फीड देंगे, then we go for the calculation of all the parameters. It may be the digestibility, it may be the any physical parameters or any other. So this is equalized paired feeding. In the slaughter experiment, usually in the when we calculate the digestibility or the retention experiments, we should go for the slaughter of the animal. This is the correct um, calculation of the retention of nutrients. How much nutrients were retained during our trial, we can only calculate to the slaughter experiments. But usually slaughter is banned for the large animal. It is also very costly. So जहाँ भी हमको slaughter करना है, there we go for the lab animal trials. Okay. So these are the 8 different type of feeding experiments which we usually conduct in our nutrition. Now about the digestibility. So the digestibility is the portion of feed or of any single nutrients of feed which is not recovered in the feces. Just we can say that amount of feed which is retained in the body. So it is the digestibility. The amount of feed which is retained in the body, it is the digestible portion, digestible part of that feed or particular nutrient. Basically, we calculate the digestibility of any nutrient. So, and any amount which is retained in the body is the digestible portion of that particular nutrient. Like if I took 5 gram of protein I have fed to the animal, and 2 gram is recovered in the feces, so 3 gram is the digestible portion of that protein. So that is the digestible portion. If I go to calculate the digestibility, then I will divide that 3 by 5 into 100. So that is the digestibility. When expressed in percent, it is known as digestibility coefficient. जब भी digestibility को हम amount में ना करके हम percent में express करते हैं, so that is the digestibility coefficient of the particular nutrient. So digestibility coefficient of a nutrient is calculated by the amount of nutrient in the feed eaten minus amount of nutrient in the feces divided by amount of nutrient in the feed into 100. So that gives you the digestibility coefficient of any of the nutrient. So how much amount of nutrient intake by the animal through feed minus amount of that particular nutrient in the feces divided by amount of nutrient in the feed. When we multiply it by 100, it is the digestibility or coefficient of a nutrient. So this is the digestibility. This is the one example I have given here. Like one bullock consumed 10 kg of grass hay and excreted 4 kg dung dry matter. Moisture content of hay is 4%. So calculate digestibility coefficient. Now students, please emphasize that here we have in example it is given जो हमें मिला हुआ है वो 10 kg grass hay means it is the actual quantity of hay which a bullock has consumed. But जो हमें dung का मिला है वो हमें dry matter मिला है, 4 kg dry matter है उसका. So हमें एक ही चीज हम लेंगे, या तो उसकी एक्चुअल क्वांटिटी लेंगे या फिर उसका ड्राई मैटर लेंगे. ठीक है ना. तो हमें एक चीज की एक्चुअल क्वांटिटी मिली है, एक चीज का ड्राई मैटर मिला है, मॉइस्चर कंटेंट यहाँ पे अपन को हे का दिया हुआ है. तो व्हाट वी डू, वी कन्वर्ट आवर क्वांटिटी एक्चुअल क्वांटिटी ऑफ हे इनटू ड्राई मैटर ऑफ हे. तो अगर मॉइस्चर उसमें 4% है, दैट मींस 96% इज द ड्राई मैटर. ठीक है. तो 96% जब ड्राई मैटर होगा तो 10 kg में कितना ड्राई मैटर होगा, इट इज 9.6 kg ना. तो 9.6 माइनस 4 अपॉन 9.6 इंटू 100 करोगे तो digestibility coefficient of the dry matter which a bullock has consumed is comes out to be 58.3%. ठीक है। तो बट दिस इज एपरेंट डाइजेस्टिबिलिटी कोफिशिएंट जो मैंने आपको अभी बताया है, 9.6 minus 4 upon 9.6 into 100, it is the apparent digestibility coefficient.